Right. My name is Brian Daniels. I am a graduate student in Robin Klein's lab at Washington University in St. Louis. And my graduate work in Robin's lab mostly focuses on blood-brain barrier biology in the context of diseases that involve inflammation of the brain and spinal cord. Primarily, I focused on viral encephalitis, so inflammation in the brain after infection with West Nile virus. But I've also had the opportunity to do some exciting work in the context of multiple sclerosis research as well. One of the key things that we study in our lab are how viruses and cells cross the blood-brain barrier and how that leads to the manifestations of disease, the damage of nervous tissue that you get from T cells, such as you have in MS. One of the key things that I, in my research specifically, are interested in is the heterogeneity of the brain, or the way that the brain in different areas differs. So you have different parts of the brain, so like the cortex or the cerebellum, or versus the spinal cord, which is another part of the central nervous system. And we know through functional studies, so from behavioral studies and those sorts of things, that these different parts of the brain do different things, but we're beginning to understand that the way that these parts of the brain interact with the immune system are also very different, and that includes the blood-brain barrier in different parts of the brain and spinal cord. And so one of the really exciting things that we've recently found in the context of MS was a gene that we've discovered regulates the blood-brain barrier differently in parts of the brain that are susceptible to multiple sclerosis, and its mouse model compared to areas of the brain that are not. And this was specifically exciting because we found this to be especially true for female mice and women with MS compared to male mice and men with MS. And so the name of this gene is S1PR2. And this study came out about 6 months ago. And so looking at the gene expression of parts of the brain, again from female mice and women who had MS, we could see the types of genes that were expressed in that tissue, so sort of looking at all the genes that were expressed there and comparing that to their male counterparts. We saw that S1PR2 was very highly upregulated or very highly expressed in those susceptible parts of the brain in female mice and women. And so through a series of studies, we showed that using pharmacological inhibition, or a drug that blocks the action of the protein S1PR2, that we could decrease the symptoms of multiple sclerosis-like disease in mice. And we also, using postmortem tissue, so brain tissue from patients that had passed away from MS, we saw much higher levels of S1PR2 expressed in female brains from MS patients compared to male brains. And so this is a first finding that we are very excited about that may explain some of the sexual dimorphism that we see in central nervous system autoimmunity. So, women are much more affected than men typically with multiple sclerosis, and so this is one thing that we found. So sort of taking those lessons though, and hopefully that will go on and possibly have therapeutic implications. But taking that lesson, one thing that I've become more interested in is just in general during inflammatory diseases of the brain and spinal cord, how do different areas of the brain differ? And so with my work with West Nile, what I'm focusing on right now are how the differences in our forebrain, so like our cortex and the parts of our brain that are very new in evolution, that sort of contribute to higher thinking and those sorts of things, compared to the hindbrain, or the parts of the brain that are evolutionarily older and that are much more responsible for autonomic functions like breathing and just sort of our basic baseline brain functions. There also appears to be evidence that the immune responses in those parts of the brain, specifically to viruses, is very different. And so using a number of mouse models again, I'm looking at the contributions of antiviral molecules, including interferons, to blood-brain barrier integrity. And so one key mystery with many of infections of the nervous system are how viruses and other pathogens access the brain, because typically they are secluded from the nervous system by the blood-brain barrier, this structure that usually keeps things in the circulation and excludes them from the brain. However, viruses like West Nile virus are able to access the CNS and cause a very serious encephalitis in some patients. And so understanding the biology of the blood-brain barrier will go a long way in helping us understand how we can prevent neurotropic or infections of the brain when you sort of are bitten by a mosquito that carries West Nile or some other encephalitic virus. And so it'll go a long way in teaching us about possible strategies for prevention, but also for medicinal interventions as well.